Hi, this is Miss Litton, and this is my wonderful, excellent Period 3 AP biologist. Say hi.
Hi.
And we are going through cellular respiration. We already had a general introduction to cellular respiration. Am I frozen? And could Blue tell Slate, what is the equation for cellular respiration? Blue tell Slate. It does. Okay, so in photosynthesis, come back to me. In photosynthesis, we took our carbohydrates. We know how to build a sugar, right? Yeah. Using energy from the sun. And we're going to do what to CO2? Are we going to reduce it or oxidize it? Reduce it. Where are we going to get the electrons from to reduce that CO2? Water. We're going to split water, and then that gives us a great byproduct, which is oxygen, which we use in what process? Cellular respiration. And we use the glucose that are made as a result of that as well, right? Okay, so what we're going to do is we're going to take the chemical bonds that are in the glucose, right, and we're going to cash those out and get it into ATP, which is the energy currency of our cell. Now, if you were hungry right now, let's say you went to In-N-Out Burger, okay? You can't take your bar mitzvah check for $5,000 and say, can I please have an In-N-Out Burger with some fries? Because they're going to go, I can't do anything with that. You have to put it in the bank. What's the name of our bank going to be? Chase. I have that one too. And Wells Fargo, which I'm sure I have like 15 accounts, right? Okay, so our bank is the bank of the mitochondria, right? Bank of the mitochondria. And it's there that we're going to ultimately use an ETC, to make some what? ATP. Good. So we have to go there, and we got to cash out our glucose in order to make some ATP. But we have an intermediary. Who is our intermediary between the glucose, before we go from here, the glucose, to the ATP? Somebody's going to hang on to some electrons for us temporarily. Say my name. NAD, you're so smart. Okay, NAD. And we're going to meet a new player, though not as popular. Okay, he's still part of the team. And does anybody know who that new player is? FAD. Oh, I love you very much. My heart is smiling. Okay? So, putting it together, glucose is going to release his electrons. And who's going to hang on to them? NAD and FAD. And we're going to take them to the top of the ETC. And we're going to na with those high energy electrons from the NAD and FAD so we can make some ATP. Now the electrons are at the bottom. Started at the bottom, now we're here. Who is going to accept those electrons at the very bottom? Oxygen forming water. Water, 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 water. That's the sixth water. Okay. Circle of life, Simba. All right, so that's why you breathe. You need something to catch those electrons at the bottom of the electron transport chain. Okay? Oxygen hooks up with a couple hydrogens and forms that water. Okay, there we go. Now he's feeling better. Okay, so when we do cellular respiration, there are several steps. Now, glycolysis, you don't even need mitochondria for that. All cells do glycolysis, and it happens in the cytosol, or the cytoplasm. And in that process, you're going to take the glucose and you're going to break it in half. Glucose has how many carbons in it? 6. So if I break it in half, I will have 2 3-carbon units, right? Okay. Guess what? One of those 3 carbons, how many steps is that going to take to do that? Did I teach you this part? It's going to take you actually 10 steps. So how many enzymes is it going to take? 10 enzymes. Guess what one of the intermediates is? PGAL. Another name for PGAL? G3P. Okay? So we're going to see that revisited, just like what plants make, right? When they're doing, what's their cycle?
Photosynthesis.
No, what's their cycle they do? What do plants do?
Calvin cells.
They do the what? Calvin cycle. What's another name for it? Light independent reaction. Old school name is dark reaction. How many times do you do that cycle? 2. Okay. We're going to do a different cycle. We're going to do the citric acid cycle or the Krebs cycle. And guess how many times we're going to do it? 2 times. Now in that cycle, we took CO2 and we reduced it into a what? Carbohydrate, right? So we were bringing in CO2 and building sugars, right? This time, what are we going to be doing? Breaking them and spitting off CO2s. You got that? Okay. So it's very similar. And then in the process, there's glycolysis. The second step is called the transition reaction or preparatory reaction. And they're transitioning into the mitochondria if you have oxygen. And then you will do your citric acid cycle, and you will write a bunch of checks. NADs and FAD checks, reduced checks, right? And we're going to take them to the top of our ETC. They'll nana make your ATP. So the sole purpose is to generate a bunch of NADs and FADs. Now, if you have no oxygen, you cannot go into the mighty mitochondria. And you're out here. And the process of glycolysis is all that you do. Without oxygen, all you get is 2 ATP every time you do cellular respiration. If you have oxygen, you could get up to 38 ATP. Maybe only 31, maybe 32. There's some debate on how much ATP, but definitely in the 30s, high 20s and 30s. If you don't have any oxygen, you only get 2. How long can you and I live without oxygen? Minutes, right? Then you're going to start passing out. If we're using duct tape, you're going to struggle, and it's going to happen even faster, right? Okay? That sounded horrible. We wouldn't, I'm sorry. Okay. And the reason is you don't have any oxygen at the bottom of your electron transport chains. So your electron transport chain, you're like, no, and it backs up. Then your citric acid cycle backs up. Your preparatory transition reaction backs up, and pretty soon you're not making any what? ATP, and you will die. Perfect. Okay, you got the big ticket items. Yes. Listen.
The reduced NAD and FAD. Do they go on the ETC?
They drop the electrons off, and then the electrons go on their merry way, and then you can reuse them again in the earlier steps. Okay, so that's like the whole big ticket. I'm just going to break down a few details for you. Yes.
What is the electrochemical ingredient to do chemiosmosis?
Where does it happen?
Yeah.
Okay, perfect. So do you remember when we did photosynthesis, we used PQ to bring hydrogen ions, right, into the thylakoid spaces? Do you remember that, the green hollow pancakes?
Yeah.
We brought it in. Well, if in photosynthesis we brought them in, what do you think we're going to do now?
Take them out.
We're going to push them out. And so in your mitochondria, you can see, and I'm thinking endosymbiotic hypothesis, are you? Is that what you're thinking? Because that's what I'm thinking. Okay, so you have an inner membrane and then you have an outer membrane. And between those two, you have what's called an intermembrane space right here that I'm highlighting in yellow. And what you do is you push your hydrogen ions to the intermembrane space. And then when you allow them to come back into the center, the matrix, that's when you make some ATP. Now remember how we use PQ? Okay, so I'm going to teach you just this part right now. Instead of coming in, we're going to go out. Go like this. Reductase. Reductase. Cytochrome oxidase.
Cytochrome oxidase.
Okay, do that part again. Reductase. Reductase. Cytochrome oxidase. So pump out, pump out, pump out. Okay? So it's going to put them all into this yellow part. When they come back into the matrix, that's where your ATP synthase complex is. You've already met him. You already know him, right? That is a protein that's enzymatic that's going to catalyze, right, the phosphorylation of ADP in order to make ATP. And you know what it's called? Oxidative phosphorylation. Why? Why is it called that? Because you're using oxygen to phosphorylate that ADP. Oxygen is the final electron acceptor. Yes?
So in photosynthesis, when you break up the water, isn't hydrogen stored in the thylakoid membranes?
Okay, so you sweep. Remember, you bring them into the thylakoid membranes, and you have other hydrogen ions as well.
Right.
okay, that you're bringing in using PQ.
So if you, and then, so when they're brought in, is that when ATP is used, or does PQ?
So when you concentrate, let's talk about that. Let's make a statement about this, because we could do a claim, evidence, reason about an electron transport chain. I think yours was the class where I recorded it in here. Was it this class? And we were talking about an electron transport chain. Okay. So when you talk about an electron transport chain, whether it's cellular respiration or photosynthesis, you could make a claim about the membrane and what's on either side. You would say on one side there are more what? Hydrogen ions than the other side. What would be the evidence for that? You have a concentration in pH, concentration in the hydrogen ions, but it's also going to affect the pH and the charge. When they're on one side, they're going to want to go to the other side. Okay? And you harvest that energy because of the difference. They go through an ATP synthase complex, and when they go through, that complex is almost like a turnstile. So as they come through to migrate to the other side, because they have a charge, and remember there's 3 ways to cross a membrane. What are the 3 ways? Through the phospholipid bilayer using a channel or a carrier or whole membrane, right? Okay, exo and endocytosis, right? Is that something that could be on your test? Is that something we talk about? Sure. Okay, could enzymes be on your test? Yeah, sure. Okay, all right, just checking. Okay, so when they go through, you harvest that energy to make ATP. And it doesn't matter if it's coming in photosynthesis, you're concentrating into the thylakoid membranes, they're moving out into the stroma, and that's where you're generating the ATP. For cellular respiration, you're concentrating them out in the intermembrane space, bringing them into the matrix, and that's where you make your ATP. Okay, more on that. Right now, you have in your brain the big ticket items for everything. Okay, can I stretch it a little bit further? Can I ask you another question? Okay, so if I was going to do a circle of life, Simba, okay, I could take CO2 and bring it to CH2O, yes? What did I just say there? Carbon dioxide to a what? Carbohydrate. What is that process called? CO2 to a carbohydrate? Oh, okay, it is a reduction, but what is the actual process called? Photosynthesis, right? Now, when you do that, right, going CO2 to CH2O, that's photosynthesis. If you go from CH2O to CO2, what's that called? Cellular respiration, right? In one of them, the photosynthesis process, right, CO2 to CH2O, you're doing a reduction, right? You're putting energy into this, yes? What are we going to use as a source for energy? The sun. The sun is going to excite electrons that you're going to send through, right? And those electrons come from water, right? When you go the other way, when you do cellular respiration, you're going from carbohydrate, you're going into what? CO2. So you're releasing energy. What are you releasing it as? ATP. So one, you're using energy from the sun. The other one, you're generating energy in the form of ATP. Yes? Okay. Remember exergonic, endergonic? All right. Okay. I just wanted to see that. And carbon is just cycling through again and again and again, just in different states. It's either in a reduced state as carbohydrate or in an oxidized state as CO2. Yes? Okay. Oh, I want to back up. That membrane thing we were talking about, claim, evidence, reason, we could claim there are more hydrogen ions on one side, right? What's my evidence? A change in pH, a difference in charge. What's the reason? Why do you have them on one side? So you can harvest that in order to make some ATP. Right? Okay, now, question.
Yes. Okay, so when you have the NAD and the FAD reduced,
yes,
and then they give the electrons to the ETC,
yes.
what is the purpose of the ETC? Just to make ATP, or?
The purpose of the ETC is this, because that electron transport chain has the ability, certain coenzymes embedded in the membrane, to move hydrogen ions to one side to another. So then you go, who cares? Hydrogen ion was. Well, we can use that in order to synthesize ATP.
Okay. But it's not for the electrons, it's for the ATP.
We use the electrons so we can concentrate the hydrogen ions so we can make the ATP. We're coupling an electron transport chain with the chemiosmotic synthesis of ATP.
Right, okay. Thank you.
Yes?
So the main purpose of the electron transfer chain is to get rid of the hydrogen ions?
The main purpose of the electron transport chain is to enable you to make a hydrogen ion gradient. So you can use that to make ATP.
Okay.
Yeah. So you're just passing the electrons along. Remember superpower, when I have an electron, reductase, reductase, cytochrome oxidase, okay? So I'm using that energy to pump hydrogen ions out because I have those electrons temporarily. Redox reactions, reduce oxidize, reduce oxidize. Then I can use that to make my ATP. Does that make sense? It's kind of like you have a dam and there's no water behind the dam, and every time you do your electron transport chain, you're dumping a bucket of water behind the dam, right? And then when I have enough water behind the dam, I can use that to do something, make some ATP. Does that help? Okay, sort of? Yes.
So in photosynthesis, would it be like a coupled reaction between the ATC and PQ?
It's still a coupled reaction between, you're doing photophosphorylation in this case, instead of oxidative phosphorylation, but you're still coupling an ETC with the production of ATP.
Okay.
Still a coupled reaction. Okay? Look at your bio buddy and say, bio buddy, which part is like, are you just grasping it? Is it a glimmer, or do you feel confident? Shadowland, sunshine? I still got to give you the details. Okay. Now, let's at this point go through the details that support this process so you can see evidence of these reactions as we go through. So the first reaction we need to talk about is glycolysis. Now, here would be the Minecraft version of glycolysis, okay? Simple, you know, really easy to follow. I'm going to come back to this. I want to show you this is closer to it, not exactly, but closer to it. I want to point out some things that you already know, love, and recognize. What molecule do we start out with? Glucose, okay? Boy, that looks really good. But you know how many carbons are in glucose? 6 carbons. So basically what you're doing here, you can see I'm glucose, and then I'm changing it a little bit, changing it a little more. Oh, look right here. Do you see how I've broken it in half at this point? Can you see that? Because now there's 2. Okay, then I'm going to take these and continue through the cycle, and take this and continue through the cycle. What I want you to see, can you see it right up here? What's getting used here and here? ATP. To get this party started, to get the ball rolling, to add a match to get our fire burning, we have to invest 2 ATP. But when we are done with glycolysis, if you can see them right here, there are 1, 2, 3, 4 ATP that are made. So I invested 2 and I got back what? 4. What did I net? 2. So am I ahead of the game? Yes. Okay. That's what you get in glycolysis. To do this process, and I know you can't see it very well, okay, and even if I make it blurry pin, my name's Blurry Face, and I care what you think. Okay, so here, okay, sorry. I'm running in the morning, I hear a lot of songs. Okay, so here's NAD, and it's becoming what? Reduced NAD. So I'm writing 2 checks. So when I'm done with glycolysis, I'm walking away with 2 ATP and 2 reduced what? NAD. Okay? That's what I'm done with when I do it. Okay? And so now that we see the more complicated one, let's go back to Minecraft. Okay? So here's our glucose, C6H12O6. It's undergoing glycolysis. There are 10 steps here with 10 enzymes that we use along the way, and you end up with pyruvic acid. Now, glucose has 6 carbon, pyruvic acid only has 3, but I have how many of them? 2. Okay? And I have to reduce some what? NAD and make some ATP. Blue, explain this reaction and what it symbolizes, because not everything is here. Go. Go, Blue.
Because in the beginning there's
Oh, yeah.
Oh, the electric.
Okay, ready? Ready, ready, ready? How many hydrogens here? 12. How many here? 8. I have 4 times 2 is what? 8. How many am I missing? 4. Who did we give them to? NAD became reduced NAD, right? That's where they went. So the energy in those bonds got given to NAD to reduce it, and now they have more energy. We have begun oxidizing the glucose, taking the energy from it and transferring it into NAD. All right? Let's learn the song. Yes, question?
To produce AED, you need 2 hydrogens?
Well, what it's written, oftentimes it's written as NADH2, or it'll just be written as NADH, but you'll know there's another electron in there, or it'll be written as NAD little r little e. Either way, you're giving it hydrogens. And it's electrons, yes. Okay, so what's the whole, did I teach you this first part of the song at all? Okay, so here we go. What's the whole thing called? Cellular respiration. Okay, so you have to learn my parts eventually too. Okay, what's the whole thing called? Cellular respiration. The first step is called glycolysis. Say it with authority. Glycolysis. Okay, who does it? And you're going to go, all cells. All cells. And I'm going to say, where did they do it? And you're going to go, in the cytoplasm. In the cytoplasm. And then I'm going to say, what do we start out with? And you're going to go, glucose. Glucose. Okay, do you see how I'm trying to make a glucose right here? Yeah. Okay, let's do it from the whole thing. What's the whole thing called? Cellular respiration. What's the first step called? Glycolysis. Who does it? All cells. Where do they do it? In the cytoplasm. What do you start out with? Glucose. How many carbons? 6. What are we going to do? Break it in half. How many steps? 10. How many enzymes? 10. What do you have? 2. So like you hold them up because it's this. Pyruvic. Now why did I do this? 3 carbons in each, okay? 2 pyruvic acids. So we remember that part. Okay, it can also be called pyruvic, okay? But either way, you're fine, okay? So from the top, what's the whole thing called? Cellular respiration. What's the first step? Glycolysis. Who does it? All cells. Where? In the cytoplasm. What do you start out with? Glucose. How many carbons? 6. What are you going to do? Break it in half. Steps? 10. Enzymes? 10. What are these called? 2 pyruvic acids. And you go like this. And you reduce some NAD and make some ATP. 2. Okay, here we go. Pyruvic acids. And you reduce some NAD and make some ATP. 2. And then pop right back here. All right, very good. Okay, that's going to become important in just a minute. All right, so now this ATP that we made, did we na-na-na-na-na it to make it? Did we use any electron transport chain? No, this is called substrate level phosphorylation. It's a phosphate getting handed off to another molecule. There's no na-na-na-na, okay? You're just taking, I have a phosphate, now you have a phosphate, okay? You cannot do this. You cannot do this unless you do this first. That's why I have it in the song that way. Reduce some NAD and make some ATP. 2. And here you're going to run into a problem. Because if you keep doing right, pretty soon all of your NAD is going to be in what state? Reduced. And you're going to run out of what? NAD, right? So you're going to have to do what to it eventually? Oxidize it. Okay, if you want to keep doing this, you've got to somehow oxidize it because otherwise you're going to run out of it. Okay, so if you look here, I just wanted to remind you about substrate level phosphorylation. So here you can see the ADP here at the top. Okay, and this molecule right here is just taking one of his phosphates and transferring it. That is substrate level phosphorylation. Okay, just a handoff. Okay, and take a look at this diagram right here. And I would like, whose turn is it, Slate?
Yeah.
Slate, it's all you. Okay, explain this diagram right here. Look at the inputs, outputs. What would be another way to say inputs and outputs? Reactants and products. Go.
So you're
So we can
Okay, I want to ask you a question. I want to ask you a question. True or false? Oxygen is a reactant.
True, yeah.
Where? I'm curious.
Oh, it's for
Okay, we lost Wi-Fi for a little bit, so I'm not sure when I lost you, but hopefully you get it. So, true or false? Oxygen is a reactant? False. Do we use oxygen at all, or do we produce oxygen during glycolysis? No. Okay. True or false? ATP is both a reactant and a product? True. Because I need 2 to get the party started, and I get 4 as a product. Yes? Perfect. Okay. Now, we need to move from this, and we're going to compare anaerobic and aerobic. Yes. Oh, yeah, I was going to answer your question. I'm sorry. Go ahead. I'm going to go back.
Yeah, please.
Okay.
Where do you get the ADP and the 4-phosphorus?
Okay. The inorganic phosphates?
Yeah.
Because you're constantly, just like you cycle NAD oxidized and reduced, you're constantly cycling ADP with ATP.
So are you like born with like a set amount and you just keep
Yeah,
working?
I'm sure you resynthesize and make more, but I'm sure there is a finite amount.
Okay.
Yeah. Okay, so now, when you compare aerobic versus anaerobic respiration, if you have no oxygen, the only ATP you get is what you make during glycolysis. So that's going to give you a total of how many again? 2. Okay? And if you have oxygen, you're going to go into the mitochondria and do those other steps we talked about, like transition and the Krebs cycle and the ETC. So let's deal with the no oxygen first, the anaerobic respiration. In this process, you're going to take your glucose, you're going to break it down into 2 pyruvic acids, you're going to reduce some NAD, and you're going to make some what? ATP. And we run into the problem that if we just repeat that step, glycolysis, 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 we're going to run out of what? Yeah, NAD that's oxidized because all of it's going to be reduced. So the second step, fermentation, its purpose, okay? People might say, oh, fermentation, the purpose is to make beer, the purpose is to make wine. It is an industry, right? But for organisms, the purpose behind fermentation is to oxidize the NAD. That's their sole purpose of fermentation. And the reason why they want to oxidize it is so they can do what again? Reduce it so they can make some ATP. So depending on what kind of organism you are, you have a different strategy for that. You and I will take our pyruvic acids, we convert them into lactic acid or lactate. What can that do to your muscles? Makes them sore, right? Ultimately, I mean, there's a series of reactions there, but ultimately it's making your muscles sore. And you notice that soreness as it builds up in your muscle maybe a couple days after a hard workout where you pushed yourself, right? And it wasn't like you fell over dead while you, oh, okay. It's not like you did that, right? But you are generating that as you went into oxygen debt because you didn't have enough. You kept working, but you're working beyond your oxygen budget. And as a result, you also made lactic acid. Now, you will convert that back eventually. Okay, that's how we solve the problem. Plants and yeast solve it in a different way. They will take their pyruvic acid and they will convert it into alcohol. Okay, and also they will release a carbon dioxide. So whereas pyruvic acid has 3 carbons in it, the alcohol they make only has 2 carbons, and they give them all off as CO2. So when yeast does that and it gives off the CO2, what does that do to your bread? It makes it rise. Unleavened bread has no yeast. Okay? Now, how come my bread, and I make bread all the time, how come it doesn't taste like alcohol? I put it in a what? Oven, which is hot. What happens to alcohol in a hot oven? It evaporates, right? Okay? But if you don't, and you take some grapes and you put it in a closed container with a little yeast or something else, and it ferments, what are you going to get? Wine. Right? Right? You're going to get wine. And if you use wheat or hops, you're going to get what? Beer. right? Do you have a highly suggested reading and thinking about that?
Yeah.
Yes, you do. Okay. So, whether you make, be with me, whether you make lactic acid or alcohol, the end result is to oxidize your NAD. Yes. I can't hear.
When would a plant have to do something with the lack of oxygen?
Okay, let's talk about that. Sometimes, now remember, plants need oxygen just like we need oxygen. Usually, they're not in short supply of it because what are they doing?
Photosynthesis.
They are doing photosynthesis. So they continue to do that. Hi, Coach. Oh. You're so cute. Thank you. Thank you. You're so sweet. You brought me a frame. Mwah. Okay. Sorry, little interruption. Okay. Yeast, plants. Okay, plants are not thinking. Okay. Oh, I can't recall. Okay, so when the plants need oxygen, they normally have plenty of it because they're doing what process? Photosynthesis generates it, right? Now, in their roots, are they doing any photosynthesis? No. So what are they only doing? Cellular respiration, right? What could happen down there if there's too much water? It could fill in all the spaces so that there's no what? Oxygen. Do you see that? So it's not going to happen all the time, okay? But it could happen that way, naturally. It could happen that way, right? Because if you flood all the air spaces, then those roots die. That's why golfers, what do they have to do every once in a while on the greens? They have to wet them. They have to aerate them, right? Do I have no golfers in this room? Oh my gosh, they have to aerate the greens. You've seen it when they do it out here, where they use a thing where they take little plugs of dirt out, or maybe your parents or somebody you've seen that in the yard when they do that, or your gardeners, okay? And they aerate the soil. They take little plugs out, and it keeps it healthy so that there are spaces so that the roots have oxygen so they can do what process? Cellular respiration. Who does cellular respiration? All cells. Where do they do it? In the cytoplasm. What do you start out with? Glucose. How many carbons? 6. Everybody be right here. There's more we're going to add on. 6. What are we going to do? Break it in half. How many steps? 10. How many enzymes? 10. What are these called? 2 pyruvic acids. And you reduce some NAD and make some ATP. 2. Good. Now pop right here. If I say to you, oh no, we don't have any oxygen, what are we going to do? You're going to say with as little oxygen as possible, fermentation. Okay? Don't tell me what that sounds like or what. I don't want. Okay? All right? Okay? Nothing. Okay, so, okay? Oh no, we don't have any oxygen. What are we going to do? Fermentation. And I'm going to say, how many choices do you have? And you're going to tell me 2. You and I make, go like this, lactic acid. Makes our muscles sore. Plants and yeast make. And you're going to come down to the other side, but you're going to take a carbon off each one. Watch, so you remember, you're going to go like this. And you wait 1 beat, and you go alcohol. And then you go either way. You oxidize your NAD. Oh no, we don't have any oxygen. What are we going to do? Fermentation. How many choices do we have? 2. You and I make lactic acid. Makes our muscles sore. Plants and yeast make alcohol. Either way, you oxidize your NAD. Why do I want to oxidize it? To reduce it to make more ATP. Perfect. Okay. Take a look at this picture right here. Maybe not this one. This is how it's used in industry. Yes? Guys, don't get loose on me. Stay. Okay? Take a look at this picture right here. Here we're going to do glycolysis. How many carbons do you see here? 6. What are we going to do? Break it in half. How many steps? 10. How many enzymes? 10. Okay. Now, when we do this, we need how many ATP to get the party started? 2. So here's 2 ATP we're investing, and you're going to get 2 ADP plus 2 inorganic phosphates. So you needed to finish that. Okay? And then ultimately you're going to make 4 ATP, so we net out how many from this? 2. Okay? And you reduce some NAD. Okay, now we're at 2-pyruvate. Oh no, we don't have any oxygen. What are we going to do? Fermentation. How many choices do we have? 2. You and I make lactic acid. Makes our muscles sore. Plants and yeast make, see the CO2 right here? Alcohol. Either way, you what? Oxidize your NAD. Okay? Yes?
Why would the lactate acid make your muscles sore?
That's another, I have a little link for you in your notes, which you can go and read about it, okay? And you can talk about it. There, I just want to continue on. Okay, so I want Blue. You please explain anaerobic respiration. Go.
That's to show that you make ATP. No, that one means you use it.
Yes, come to me. Here we go.
What is the acetaldehyde?
What?
Like I understand that ethanol is the alcohol, but what is the acetaldehyde?
It's just a transition. There's like more steps than I'm showing you, so I'm just showing you kind of the beginning and the end.
Okay.
So that's just a transition in it. Okay, Blue, you just went, yes? Yeah. Slate, this is yours, go.
Fermentation inputs which is that
All right, be yourself. Here you go. Be yourself. Love yourself. Let's go. Log in. What was your question? Ask me your question.
So, if you concentrate all the electrons or hydrogens on one side of the brain, how
I'm not doing a membrane yet, so you're gonna hang on to that. Let's go. The rest of you need to log in. Two more of you need to log in. Let's go. Waiting on 2 of you to log in. All right. Oh, yeah. Sadness. Good. Oh, my God. Oh. Okay. So which of these is not true? So that means whenever you see a question like that, you assume all are true. And you just have to find the lie. Right? Is there a net gain of only 2 ATP? Yeah, that's a truth. Does it occur in the cytoplasm? Yes. Do we have an electron transport chain? No. There is no none-a-none-a-ness. Okay? We didn't. We did substrate level phosphorylation. We did begin with glucose, and it can happen with yeast. Okay? So that's that one. Ask your bio buddy, Shadowlands or Sunshine? Clarifying questions to ask me. Oh, this one, hang on a second. This one doesn't want to start. So sometimes when I start one on another page, it will work.
true?
The answer is true.
someone actually answered.
Good job. Oh, it still didn't do it. Okay, so go. Okay, go ahead and talk about this one with your bio buddy since I'm not having it. Let me see if this will start it. Oh yeah, this will. Now watch, it'll work now. Do you want to stop the current? Yes. Okay, now it'll work. It's I don't know why this question's like this. Here it comes, here you go. It will. Just trust me. It will.
There you go.
It's done now. No, it's not.
It will.
Okay, there it is.
Ah.
You submit? How? How did Michael make that up? No, but he's like,
Michael made a deal with the devil and the clickers. He sold his soul to be first in all clicker clubs. Is this a man?
Yes.
Are multiple answers necessary? Yes, I've told you it's possible, but it is necessary. All right. So before I hit stop, which are the possible products of fermentation? Is lactic acid a product? Yes. Who makes that?
We do.
We do. Is glucose a product? No. What would you consider it? Reactant. Is alcohol a product?
Yes.
Who makes it?
Plants.
Denise. Is carbon dioxide a product?
Yes.
Who makes it?
Whoops.
Plants and yeast. Is oxygen a product? Is it a reactant?
No.
No. It's neither. Is it acetyl-CoA? I haven't even taught you that.
come on.
No, we all got it.
Oh, Lori. There you go. Where's Michael? Where's Michael?
Oh my God, Michael.
Oh my God. All right.
True.
False. True. Okay. Now, this is our put it all together once again. Style it in, guys. Right? I have like maybe 22 minutes with you. Okay. So we want to make sure we stay focused. We look right here. We're going to talk about aerobic respiration now. Okay. And why we want to do that. So here in glycolysis, it has like stutter to pyruvate, but we really know how many steps is it? 10. How many enzymes? 10. Good. And when we are done, we will have these 2 pyruvic acids, and if we have oxygen available to us, we're going to transition. This step right here, if O2 is available, is called transition or the preparatory reaction. Okay? And we're going to move on into the mitochondria. Now, we have these NADs that we made. Reduce some NAD. Look at me. Reduce some NAD and what? Make some ATP. These NAD, we don't need to send through fermentation anymore. Because what do we have available to us? Oxygen. So we're going to take them eventually to a what? ETC. Okay? Not yet, but we'll get there. Okay? When we do transition prep, we're going to make some more NADs that can go to our ETC. And when we do our Krebs cycle, okay, instead of like in the other one where we were taking CO2 and reducing it, right, we're going to be oxidizing it, and this Krebs cycle is going to be cranking out some carbon dioxides. Okay? And ultimately, between transition and Krebs cycle, how many CO2s will we crank out? 6, exactly. Okay. Why 6? C6H12O6. Okay, so we're going to crank out 6 CO2s, right? 6 carbons in, 6 carbons out, right? Okay, and the C6H12O6, the glucose just got oxidized, right, into CO2. What's missing from our glucose here? Hydrogens. Who are we giving them to? NADs, so we can take them to the top of our ETC. Make some ATP, right? Okay. Bring it, putting it all together. Okay. Who is going to be the final electron acceptor so we can continue to make this ATP? Oxygen, which is going to hook up with some of those hydrogens and make some water. And those two circle back and forth, right? Okay. So, let's talk about the structure of the mitochondria. Okay, the innermost part right here is called the matrix. You're going to do your Krebs cycle here, and your electron transport chains are happening all on this inner membrane right here. Those are called the cristae, those little folds. Okay, and let's see if you can do this. Yeah, I told you it's a map, but it is a man. Where does glycolysis occur? Does it occur in the mitochondria? No, I just talked about it, but it doesn't occur there. Does it occur in the cytoplasm? Yes. Who does it?
Oh my god.
Does it occur on membranes? No. Does it occur in chloroplast? No. Oh my God.
Go home.
Now, listen to me. I asked this question right here to serve a purpose. You have got two huge metabolic reactions in your head right now. Photosynthesis and cellular respiration. You have to keep them straight. Think beginning, end. What am I doing? What organelle am I in? So you don't miss questions like this. Because the more we learn in biology, the more these, okay, you're going to know stuff about them. You know things about all of this. And so just because there's a familiar word down here that you know about it doesn't mean it's the right answer. Okay? Before you had acetyl-CoA in that other question, you're like, never even heard of that. Still some of you picked it, okay? But you're going to start to know some of these. So you got to think through the big picture when you're answering them. Try again.
Oh, Michael already knows yes and no. I'm giving up.
Think. Because I'm lame. If you need it. Oh, he doesn't.
Wait, it's A and E, right?
All right, let me start talking to you about it. Okay? For glycolysis, do you need oxygen? No. Do you need glucose?
Yes.
Do you need NADOX?
Yes.
Yes. What are you going to start out with? If you're going to reduce some NAD and make some ATP, it's got to be what first? Oxidized. Do you need NAD reduced? No, it's a product. Do you need oxygen? No. Do you need mitochondria? No. Do you need ATP? Yes. 2 to get the party started. Do you need pyruvic acid? No. It's a what? Product. Oh, Michael. All right. Okay, here we go. Let's try again.
Oh my god. Wow.
What? Use that large mammalian brain.
I forgot about
I have like math expo tomorrow.
Don't try. All right, I want to start working through this. Products of glycolysis. Glycolysis, is oxygen a product? Yes or no?
No.
No. Glucose? No. No. NADOX?
No.
No. NAD reduced? Yes. Is oxygen? No. How about mitochondria? No. ATP a product? Yes. How many ATP do you make as a result of glycolysis? 4, but we net out 2. Okay, is pyruvic acid a product? Yes.
God.
What is Michael? Nowhere to be found.
Oh, Nick! All right.
Michael's not even on the food.
All right. Here we go. Now, we just finished glycolysis. Tell me right here. Reduce, be right here. Reduce some NAD and make some ATP. 2, be right here. Okay? Now, if we have oxygen, I'm going to go, oh, we have plenty of oxygen. What should we do? You're going to say as breathy as you can, opposite of fermentation. You're going to say aerobic respiration. Aerobic respiration. Okay, good. And then I'm going to say, what's the next step called? And you're going to go like this. Transition prep. Perfect. Okay? Because we're transitioning into the mighty what? Mitochondria. The what of the cell? Power of the cell mean. Okay, now, people, stay with me. Okay? Look right here. Look. It's okay. Your songs you can stop being pyruvic acid. Okay. Here is pyruvic acid. How many of these do I have? 2. How many focus here. How many carbons in each? 3. 1, 2, 3. Yes?
Yeah.
Through the process of transition prep, we're going to take this pyruvate and we're going to take this coenzyme, and he is going to be the one that escorts it into the mitochondria. Okay? So there's 3 carbons all hooked together. He says, I'll escort you in, but you got to ditch one of those guys. Oh, okay. So you get 1 carbon that's ditched. He goes, okay, let's go on into the mitochondria. So you can see this CoA that's right here, okay? This CoA that's right, oh, that's too dark. I'm so sorry. This CoA that's right here is now right here. And here are still our 2 carbons. Where is the 3rd carbon? Where did it go? Right here. 1, 2, 3, right? Here were the 3, here it is. It's now attached on there. Now, I want you to see this. This is CO2 right here, yes? See the CO2? Do you see the CO2? Okay, now, what is missing? The what? Hydrogen. Where is it?
NAD. Reduce.
Further oxidizing the glucose, right? We're in the pathway. Blue, explain this reaction right here. I've already Instagrammed it. Go. Okay, come back to me. We're right here. Ready? We have plenty of oxygen. What are we going to do? Aerobic respiration. You say it with authority. Go. Engage. We have plenty of oxygen. What are we going to do? Aerobic respiration. That was way better. Okay, what's the next step called? Transition prep. And you go like this. Here we go. Now watch me. Watch what I'm going to do. What did I just do? I gave off a carbon as what? Carbon dioxide. I'm reminding myself of that because I'm going. So now I'm here, yes? And you go like this. And you. Reduce some NAD. Now these are called acetyl-CoA. Acetyl because of the 2 carbons, and CoA because it's a coenzyme. Okay? So, and you reduce some NAD and 2 acetyl-CoA, put 1 away. Okay, here we go. Here we go. And you reduce some NAD and 2 acetyl-CoA, put 1 away. The reason why I'm putting 1 away is I'm going to take this guy and feed him through the Krebs cycle, and then I'm going to whip out this guy and I'm going to feed him through the Krebs cycle. See why I'm putting it? Okay. All right, whole thing so far from the top. Here we go. What's the whole thing called? Cellular respiration. What's the first step called? Glycolysis. Who does it? All cells. Where do they do it? In the cytoplasm. Good. What do we start out with? Glucose. How many carbons? 6. What are we going to do? Break it in half. Steps? 10. Enzymes? 10. What are these called? 2 pyruvic acids. And you reduce some NAD and make some ATP. 2. Oh, we have plenty of oxygen. What should we do? Aerobic respiration. What's the next step called? Transition prep. Here we go. And you reduce some NAD and 2 acetyl-CoA, put 1 away. Awesome. Now, we made 2 NAD in glycolysis, right? We made 2 NAD just now. We can take all those NADs to the top of the ETC, but we're going to make some more first. Okay? Make some more first. So now, the next step is called the Krebs cycle or the citric acid cycle. Ladies, it's called the citric acid cycle because the first thing you make is called citric acid. okay, or citric, okay? So that's the first thing you're going to make. This is the one that's in your book. I find this not helpful, so here's the one I made, okay? All right, so hopefully this makes it a little bit easier. You ready for this? 2 acetyl-CoAs. Now I have just written it as C2. You know it actually looks like this, right? This is the 2 acetyl-CoAs that are right here, yeah? And how many carbons do they have? 2. So that's why I'm just saying here, C2. This cycle is easier than your other one, just in case you're wondering. Don't like, okay? Now, remember how when we did cellular photosynthesis, how it was preloaded with RUBP, C5? Okay? This is also preloaded with a what? C4. We're going to take a 2 carbon, we're going to make a 4 carbon, and we're going to put them together. What are we going to get? 6 carbon. Now that we have this 6 carbon unit, we're feeding in acetyl-CoA, a 2 carbon. We're going to pull off a CO2, and in the process of pulling it off, just like in transition prep, okay, when we pull off that CO2, we can also reduce an NAD. So we had 6 carbons, we pulled one off. What do we have now? 5. You all right? We're going to do the same thing all over again. Pull off a CO2 and reduce another NAD. Now we're down to what? C4. C4. Now I have that I can take to the ETC, and this I can take to this ETC. You with me? I'm taking the energy out and giving it to the NADs by reducing it. Now, to help us remember this, okay, we're going to go like this. Whenever we do this, this is a litinism. This is nothing real. We're going to say that's do the bit. So we're adding a 2 carbon to a 4 carbon. Listen to me. 2 plus 4 is 6. You hearing it? Do the bit. Get 5. So we took 1 away. And then we're going to do the same thing all over again, and we get what? 4. Okay, here we go. Watch me. 2. You can get with me. You'll figure it out. 2 plus 4 is 6. We always do 6 like this, right? Whenever we make 6, we're like, thumbs up. Yeah. Okay, here we go. 2 plus 4 is 6. Do the bit, get 5. Do the bit, get 4. That's super easy. Way easier, right? Go again. 2 plus 4 is 6. Do the bit, get 5. Do the bit, get 4. Okay, look what's happening here. I see a little substrate level phosphorylation. Okay, you're gonna make, and what are we making right here? ATP. We're going to reduce nuclea. You already told me about them. Reduce an FAD. Reduce an NAD. Now notice we kept our C4 in this, right? The whole time through. Now he's preloaded. And we'll take our second acetyl-CoA. 2 plus 4 is 6. Do the bit, get 5. Do the bit, get 4. And then you're going like this. Make an ATP, roll it forward. Make an ATP. Now take it the other way. Reduce an FAD. Reduce an NAD. And 2 plus 4 is 6. Do the bit, get 5. Do the bit, get 4. Roll it. Make an ATP, reduce an FAD, reduce an NAD, and 2 plus 4 is 6. Do the bit, get 5. Do the bit, get 4. Make an ATP, reduce an FAD, reduce an NAD, and stop. Gather all your reduced NADs and FADs. Say that with me. Gather all your reduced NADs and FADs and take them to the top of the ETC. And take them to the top of the ETC. Now, okay, you're right there. You're right there. Okay. Now, let me talk to you about when you take them to the top of the ETC. Do you remember when we learned about na-na-na-na-na-nas? Yes. Okay. Now, some NADs are really powerful and strong, so they literally get to come in the top. So they na-na-na-na a lot. So they make a lot of what? ATP. But some are not as powerful, and they come in a little bit lower. Let me tell you who's not powerful. The NADs in glycolysis They're way out in the cytoplasm. They got to all come all the way in here. They lose a little power, or they hand it off to FADs. So they come in a little lower. FADs also come in a little lower. So they don't get to na 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 na. They just get to na 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 na. Okay? So they don't make as much ATP. So when I say take it to the top of the ETC, you're going to go like this. Because some come in at the top and some come in part way. Okay. Gather all your reduced NADs and FADs and take them to the top of the ETC. Then you know this part, what's going to come next. Reductase, reductase, cytochrome oxidase. Okay, do that again. Reductase, reductase, cytochrome oxidase. And you make some ATP. Now, how much? You ready? When you come in strong, you usually make about 3 ATP because you got reductase, reductase, cytochrome oxidase. I'm cranking out hydrogen ion, hydrogen ion, hydrogen ion, right? But if I come in a little lower, I miss the first one. I'm just reductase, cytochrome oxidase. So I only make how many? 2. So you go like this. And you make some ATP. A lot of it, a lot of it, a lot of it, or a lot of it, a lot of it. Okay? So how many times did I say it the first time? 3. How many times did I say it the second time? 2. Gather all your reduced NADs and FADs and take them to the top of the ETC. Reductase, reductase, cytochrome oxidase. And you make some ATP. A lot of it, a lot of it, a lot of it, or a lot of it, a lot of it. Who catches the electrons at the bottom? Oxygen forming water. Water, 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 water. What are we going to do? Break it in half. What do we get? Oxygen and electrons. Sweep, sweep. Here comes the sun traveling in waves. Particles of light are called photons. And it gets the electrons excited. PQ, and you make some ATP. Here comes the sun traveling in waves. Particles of light are called photons, and it gets the electrons excited. Who catches it? NADP, forming reduced NADP. Then you take some ATP and some reduced NADP to the dark reaction, or the Calvin cycle, or the light-independent reaction. 3C5 plus 3C1 makes 3C6. Break down to 6C3. Use some ATP. Use some reduced NADP on your 6C3. Take 1 away, PJL or G3P, and you're left with 5C3. Use some ATP to build 3C5 plus 3C1 makes 3C6. Break down to 6C3. Use some ATP. Use some reduced NADP on your 6C3. Take 1 away, glucose, jazz hands, and you're left with 5C3. Make some ATP to build 3C5. Stop. Pick up the glucose. What's this step called? Glycolysis. What are we going to start with? Glucose. How many carbons? 6. What are we going to do? Break it in half. Steps? 10. Enzymes? 10. What are these called? 2 pyruvic acids. And you reduce some NAD and make some ATP. 2. Oh, we have plenty of oxygen. What should we do? Aerobic respiration. What's the next step called? Transition prep. Here we go. And you reduce some NAD and 2 acetyl-CoA, put 1 away. What's the next step called? Krebs cycle. What's another name for it? Citric acid cycle. How many times are we going to do it? 2. Why? 2 acetyl-CoA. Every time we do the bit, what are we doing? Reducing an NAD and pulling off a carbon dioxide. Ready? Here we go. 2 plus 4 is 6. Do the bit, get 5. Do the bit, get 4. Make an ATP. Reduce an FAD. reduce an NAD, and 2 plus 4 is 6. Do the bit, get 5, do the bit, get 4. Make an ATP, reduce an FAD, reduce an NAD, and stop. Gather all your reduced NADs and FADs and take it to the top of the ETC. Reductase, reductase, cytochrome oxidase, and you make some ATP. A lot of it, a lot of it, a lot of it, or a lot of it, a lot of it. Who catches electrons at the bottom? Oxygen forming water. Yay! Okay,
I'm so happy.
Makes you happy? Okay. Here we go. Come back to me. Okay. So, look right here. So here's the top of the ETC. This is that cristae, the inner folds of the mitochondria, right? Okay, so we're going to make the electrons go through reductase. Reductase, what? Cytochrome oxidase. And you make some ATP. A lot of it, a lot of it, a lot of it. Or if you only came in part way, here's FAD, a lot of it, a lot of it. Got that? Here's another picture. Here. Reductase, reductase, cytochrome oxidase. Do you see it right here? But FAD is coming in right here. You all right with that? Now, here's the deal. You could, where is this? Here. Catabolism and anabolism. Okay? Catabolism is about what? Breaking, and anabolism is about building. Okay? Now, riddle me this. You don't have to just use glucose. You can feed in fats and you can feed in proteins. Think this through. What has more calories, carbohydrates or fats? Fats have more calories than carbohydrates. Because for each carbohydrate, for each glucose molecule I have, I'm breaking it in half, and I'm making how many pyruvic acids? 2, which will make how many acetyl-CoAs? 2. With a fat, remember the glycerol and the long fatty acid chains? Each glycerol will get me an acetyl-CoA. And let's say I have a fatty acid chain that's 18 carbons long. Remember saturated, unsaturated? I have 3 of those that are 18 long. Snip them into 2s for acetyl-CoA. Each chain can get me 9 acetyl-CoAs, right? And how many chains do I have? 3. What's 3 times 9? 27. Plus I get a 28th acetyl-CoA from the glycerol. When I burn glucose, I only have 2 acetyl-CoAs, but I could have 28 acetyl-CoAs from a fat. So do you understand why fat has more calories than a carbohydrate? Okay, are you seeing that? Because it'll generate a lot more ATP rapidly. Okay. Why do we burn? It's good for you. You need it for other things too. Got to have fat in your diet every day. Okay? No eating disorders. Okay. This I have already put up. Okay, just comparing the 2 chloroplasts and mitochondria. We've done that several times. Okay, that's just a scary diagram. We don't have to pay attention to that. But this I want to talk to you about. Okay, now go in your notes quickly because we still have our quiz too. Okay, skip to the very end and then we'll fill in all the ends. Go to the very last box of your notes. Are you the very last box? Yeah. This is going to loop back to the first chapter we talked about in this unit. Okay, look at this word, phosphofructokinase. That's a nice hangman's word. PFK is an allosteric enzyme. Allosteric enzyme. Think about that for a minute. Allosteric. Remember the secondary site, the binding site? Allosteric enzyme active early in the pathway of glycolysis, and it is what? Inhibited by high levels of ATP. This in turn is going to stop glycolysis. Now think about this. Remember when we talked about glucose, what are we going to do? Break it in half. How many steps? 10. How many enzymes? 10. Here's one of your 10 enzymes. And it is inhibited by the production of ATP, which is the product of this whole reaction, and we're stopping it way back in glycolysis. What kind of inhibitor is it if it binds to an allosteric site? If the ATP binds to a non-competitive inhibitor. You see this?
Oh yeah.
This, then you can say now, okay, that it is the pacemaker of cellular respiration. If I have plenty of ATP, do I need to do this reaction? No. If I don't have any ATP, do I need to do this reaction? Yes. And it is available to catalyze those reactions. Ah, okay, got it? Okay, let's fill in those notes and then we'll take our quiz. Yay! Okay, so which part in 8.1 was there anything we missed there? Oh, and hey, we hope you're having a great day. We're going to sign off now. You're super smart.